You've already done this in your simulation last week or the week before, depending on which group you're in, but it's pretty straightforward. We have our plate here. We draw a T is what we usually do. You can do more quadrants, but 3 sectors is fine. You draw that on the back, on the bottom, right? That's where all of our writing goes. Then you flip it over. You get your culture, okay? Once you have your culture, first, you incinerate your loop, right? Make sure that it is sterile. And with this, again, because we're dealing with a broth, swirl it around a bit, make sure that it's all mixed up, and you can go straight in and not worry about killing anything. You swirl your loop around just to make sure you get plenty of stuff on there. And then, let's see if we can do this in a way that you can see. Terrible. Be right back. All right. So we have our loop that has culture on it. We have our plate here. Okay. First thing you do is you lift the lid just slightly. Of course, I have to lift it off more so you can see what's going on. And you run it over this first sector. Like that. Okay. Just spread it out. You can do both sides of the loop. That's it. Only the first sector, right? Remember this from the online lab. Okay, you go back, you incinerate the loop again. It doesn't take long. Now, at this point, you have a hot loop. You don't want to wave it around to make it cool down, so instead, you take it and you touch it to a spot that you have not yet inoculated. Okay, that cools it down, and then, and again, I'm taking off the lid so you can see what's going on. You just grab a little bit from the first sector and you bring it into the second. Don't push hard. Don't use the tip. You will gouge into it. You don't want to do that, right? So you spread that around. Make sure that that's nice and spread. And then go back, incinerate your loop. And of course, again, while you're doing all of that, you keep the cover on this. Then, again, you touch a spot that you have not yet inoculated. Not sure if you heard the little sizzle there, but you can certainly see where it melted because the loop is hot, right? If the loop is hot and it touches live bacteria, those live bacteria ain't live no more, right? You want to spread these bacteria out so that you can grow them into individual colonies. They have to be alive to do that. So, now you go up to your second quadrant, and again, you just drag down a little bit, and you spread it, streak it over the third one. Now, when you do that for this, it's best not to overlap, okay? So you take it down, and then once you have it here, you just go side to side and make a sort of like Z pattern or Z pattern, back and forth, back and forth. You don't want to bring it back up because the idea, again, is you're trying to spread things out. And as you move it along the plate, more and more of stuff that's on the loop will go onto the plate, hopefully spreading it out so that after you incubate it, you'll have nice separate colonies. Once you have all 3 of those sectors done, you lower the lid again, you put the lid back on, whatever, and you put it upside down. And when the time comes, we'll put that in the incubator and we'll be good to go.